Welcome to this episode of Life Science Success. For those of you who don't know me, my name is Don, and I'm a digital marketer in the life sciences space. And today we are going to talk to Dr. David E. Martin. But before we get to that, we have a quick word from our sponsor.
Episode of Life Science Success is brought to you by D3 Digital Media Marketing. Dive into the digital world with confidence and creativity on your side. D3 Digital Media Marketing is your all-in-one solution for making a lasting impact online. From the vibrant, engaging world of social media marketing, podcasts, and webinars, to the strategic depths of website development, SEO, pay-per-click, email, and content marketing, we tailor our approach to fit your unique brand story. Let's elevate your presence, connect with your audience, and drive success together. Your digital journey starts here. Visit d3digitalmedia.com to learn more.
So with that, let me just quickly introduce Dr. David E. Martin. He's the president and CEO of TrippBio, a distinguished translational scientist with over 30 years of experience in drug development. Dr. Martin has led numerous groundbreaking projects and is here to share his insights on the future of therapeutic advancements. So with that, welcome to the show.
Thanks, Don. It's great to be here.
Yeah, thanks a lot. I really appreciate it. And, I'm excited to sort of dive a little bit deeper into what you're doing at TrippBio. But before we get there, can we just talk a little bit about you and sort of what is your background and what brought you into life sciences?
Sure. So I am trained as a pharmacist. I got my pharmacy degree from the University of Southern California. I started in the late '80s and graduated in '91 in Los Angeles. Just as the AIDS epidemic, also then known as GRIDS, was starting to come about, and I'd gotten very interested in that disease. It was quite interesting. There wasn't a lot known about it. I was an intern pharmacist at Huntington Memorial Hospital, and we had several people come through. Atypical. At the time, it was supposed to be a gay disease, which is why it was called GRIDS. And the people that I was seeing in the hospital weren't homosexual. They weren't. Most of them were either white or Hispanic. They'd gotten infected through blood transfusions or just through normal heterosexual activity. And it was different than what the news media had been talking about at the time. And that got me really interested in infectious diseases in particular. And during school and my postgraduate training, I got really interested in clinical research. I really liked the idea of, asking a question that no one else had ever asked, and then trying to figure out how to answer that question. And it was interesting. It was new knowledge. It was something that no one else was doing. It was amazing to me that there were actually things that no one much smarter than myself had never asked. And sometimes that related to clinical things that we were seeing in the hospital. Sometimes it was with new drugs that were starting to come through the pipeline. And I was fortunate to have a very good mentor, Dr. Jack Kern, who really nurtured that scientific curiosity. He continually asked why. I would say something and he'd say, yeah, but why is that? Why does it do that? And it got a little annoying at first. But then I figured out why he was doing it, the why again. And it really became sort of the key underpinning for all of my training. Why is this? Why have we always done things this way? Why can't we do something else? Why can't we do it differently? Why do we have to do, this type of an approach on that type of activity? And it just, that really got my scientific curiosity primed. And I've had a very interesting career, 33-year career now in clinical research that's been just, countless questions asked, and most of them answered, some of them not. Some of them answered in ways that I never would have anticipated. and some of them answered in ways that were heartbreaking.
Uh,
But that's just the way science goes sometimes.
yeah. It is one of my favorite sections in most academic papers. Whenever you get to the very bottom, and there's like, hey, these are all the things that are left like unanswered
Right.
that still is open for the next researcher to come along and sort of ask questions on. And I sort of enjoy that. Not necessarily that I'd you know maybe enjoy the same level of somebody asking me why all the time, but yeah, I certainly do enjoy that aspect of hey, look, as a researcher, here's as well, here are all the things that we didn't find in our research so far.
Right. And a lot of times what you'll also have is there'll be you start out with an experiment, and it'll be successful, but it'll be successful in a way that you didn't anticipate. Because I tell people all the time, well, this is why you do research. If you knew the answer, it would be called confirmation and not research. And so sometimes those paragraphs also have, well, here's what we found different than we anticipated,
Mm-hmm.
and then that leads, here's why there are these series of next questions, and it's trying to figure out the puzzle pieces in the research sometimes that can be quite interesting. And that's one of the reasons why I like translational sciences, going from animals to people, because no animal model is perfectly predicts the human response. There's always something different that happens in that translation phase, in that translation process, and it's incumbent on the scientists to figure out, okay, what are those differences, and are those differences meaningful? Sometimes they are, sometimes they're not. Sometimes they're just a nuisance, but sometimes they can be quite devastating
Yeah.
if they're not followed up properly.
Yeah. Yeah, and just so many good points as well that you're making, that hey, look, sometimes those unanswered questions or the next steps could lead you to something else that either sort of says, hey, look, this is not going to work out, or maybe there's some other boundary that you haven't considered as well, and so just absolutely critical.
Right, right.
As you've gone through your career in drug development, though, can you talk about some of the highlights that you've had in your extensive experience?
Sure. So one of the projects that I got involved with as a clinical research fellow at the University of North Carolina in Chapel Hill was the second year of that fellowship I spent at Glaxo. And this was before it was Glaxo Wellcome or GlaxoSmithKline. It was just Glaxo. And we were working on the phase 3 program for lamivudine, which was their nucleoside um, analog for treatment of HIV infection. And at the time, this was back in the early to mid 90s, we were still doing treatment studies with a clinical endpoint. And so you had to had to study patients all the way to either an AIDS defining event or death. And, sometimes if you got patients that were late enough in their infection process, um, those endpoints came fairly quickly. If you got them early enough, it could take years to get the right number of endpoints to be completed. And so, one of the things that everyone was looking for were surrogate markers, some way to shortcut the time to get to a clinically meaningful endpoint, but without changing overall dynamics and trajectory of the therapeutic benefit. And so we got involved in trying to validate this new technology called PCR. And we were looking at HIV RNA. And, this was before we knew much of anything about it. This was, back in the early days of PCR, where, it was incredibly finicky, very difficult to work with, very challenging. And we thought that there was going to be a relationship between the amount of HIV RNA circulating and a patient's progression to disease, to those endpoints. That the higher the value of HIV RNA, the sicker the patients were. because it looked at the amount of replication that was ongoing in an individual. And so the problem was no one was collecting RNA in their clinical studies, and no one had ever collected RNA in a clinic before. And so we had to figure out kind of how to do some of this. And we got together with the VA cooperative group out of New Haven, Connecticut, and they had conducted a study back in the late 80s looking at efficacy of AZT at the time in a VA population. And they had stored samples that they had kept in freezers. And it was a relatively large study with relatively large number of endpoints. And so we were able to take those samples and, again, not knowing a whole lot about HIV RNA at the time, not knowing if we would even be able to detect it in those stored samples because they hadn't been stored and processed properly. So we ended up doing that, and turned out that the RNA was relatively stable over time, stable in the samples that we had collected, or they had collected. And we completed that analysis and found that, yes, in fact, the amount of HIV RNA was related to disease progression, and patients that were treated that had a decrease in viral load tended to do very well, and their progression was delayed. And so that was really the first study that was confirmed that HIV RNA could be a surrogate marker for disease and could be used in a drug development program as a potential endpoint in those studies, replacing the hard clinical endpoints of hospitalization or death, or progression to AIDS from HIV infection. And that became really the basis for the FDA agreeing to accept HIV RNA as a surrogate marker. It wasn't the only thing that did it. There were John Mellors and his group published some work on HIV RNA that helped as well. But we were really the first study to show that drug treatment could positively impact HIV RNA levels. And I did that as a fellow, and being involved in such cutting edge research at the time, seeing data that no one else had ever really seen before, really I think is one of the things that got my appetite whetted for doing this kind of research. And then a decade later, I was involved in a company called Panicos Pharmaceuticals, where we were working on a drug that had a very unique mechanism of action, one that we had never seen before. And it took us a little bit of time to figure out what it was, but it was completely unexpected. It was a serendipitous finding for a new class of drugs called maturation inhibitors. And it all came about because of an academic researcher using a replication blind screen to essentially look at plant extracts to see if there was any HIV activity. He found an extract that seemed to have activity. He then went in and deconvoluted the sample, figured out what the active compound was, and then, without knowing anything about the mechanism, he took that compound and did medicinal chemistry around it and created a more potent compound. Again, without knowing how it worked, all he knew is it inhibited HIV RNA or HIV replication in the test tube. And so we had the rights to that compound, and it was our job to figure out how it worked. And I'm not a virologist by training, but I worked with some world-class virologists, and we figured it out. And it turned out that it was binding not to an enzyme, not to a receptor, but actually binding to the GAG polyprotein at a very specific place that was only accessible during a certain point in the viral life cycle. And so the drug became attached to the GAG SP1 cleavage site. And that's the last place that the protease enzyme cleaves prior to forming a fully mature infectious viral particle. And again, it's the very last step in viral replication. And because it's a small molecule bonding to a protein that's inhibiting, and it's that bonding that inhibits the protease from accessing that cleavage site. No one believed us. Everybody was like, "No, you're wrong. It's all about, it's an atypical protease inhibitor. It's got to be." And so it took us a fair bit of time to work through that. We were able to take the compound through preclinical studies into the clinic. We confirmed that it had HIV RNA activity, and it was very potent. But the compound was flawed, and we ended up having to kill that particular program. But there are still maturation inhibitors in the clinic. GlaxoSmithKline has a program that's now in phase 1, moving into phase 2 shortly. I'm still working with another group of investigators on maturation inhibitors, 22 years later. So I definitely believe this is a class that's worth trying to advance. But those are, just a few of the questions that, are unexpected.
Yeah.
And I think my experience with Panicos in that novel therapeutic area has really informed a lot of the work now that I'm doing at TrippBio in terms
Perfect.
of a novel target and, people not believing the data.
That's actually a perfect transition, because my next question leads us to the point of, so what essentially led you to the point of becoming the president and CEO of TrippBio?
So it was a relatively short path. So I spent about 30 years at the time. 30 years in drug development. I'd been CEO of another company, a small biotech that I'd started, and that was, we had licensed a program, raised money, designed a killer animal experiment, no pun intended, that really was designed to say, is this drug going to work? Yes or no. And unfortunately, that was one of those crushing answers that no, it didn't work. And I was looking for additional opportunities at the time, and so this was in early mid 2020, and I got involved at that time as a member of the board of directors with TrippBio. Um, they were looking for board members with my type of experience, virology, drug development. And the science and the technology I thought was quite fascinating. It had all come out of Dr. Ralph Tripp's lab at the University of Georgia in Athens. Um, about the early 2010s, he had conducted a series of experiments looking for host targets that were involved in viral replication for influenza. And he had conducted a fairly extensive siRNA screening um, effort to look for host targets that were, again, directly involved in viral replication. And he found one that seemed to be incredibly important for viral replication. And it was a protein that encoded for an enzyme called OAT3, the organic anion transporter 3 enzyme. And so when he finished off his experiments, he found that, yes, did that particular enzyme did appear to be incredibly important for viral replication. Not only if you knock it out, but if you pharmacologically inhibit it with a very potent inhibitor, you'll see great activity both in vitro and in vivo. And the compound that he used was probenecid, which is a prototypical OAT3 inhibitor. So he published that work, and then it just sort of lay there for a while until the SARS-CoV-2 virus reared its ugly head. And they're very similar viruses. They're both negative strand RNA viruses, respiratory with a focus. And so he thought, well, maybe probenecid would have some activity. And sure enough, it had very potent in vitro activity, very potent in vivo activity. So the university filed a method of use patent that became the foundational IP for TrippBio. And that approach was very interesting to me. And so I joined as a member of the board of directors. And about 9 months later, we had a CEO transition, and the board chairman asked me if I would take over, and I eventually said yes because the data was just so compelling. Um, the data with influenza was incredibly exciting. Picomolar activity. The data with SARS-CoV-2 was very good. Um, I knew pharmacologically that we were going to be able to achieve, very high plasma concentrations. So we were pretty guaranteed that we were going to see some activity. And we were starting to move into exploring the in vitro activity against respiratory syncytial virus, or RSV at the time. And the data from a virology standpoint was just too good to pass up.
Mm.
The in vitro potency was solid. The in vivo activity looked incredibly good. We were seeing significant reductions in viral lung titers across all the viruses. And so to me, it was like, yeah, this is going to work. Forget what the drug is for just a second. If you look at the virology data, it's outstanding. Um, and so that got me involved as the CEO at the time. So we were able to raise money, conduct a clinical trial with COVID-19 patients where we showed significant activity. And now we're starting to expand into additional RNA viruses.
So I follow Dr. Eric Topol pretty closely on Twitter, and I mean he's constantly talking about new variants and sort of new progression for this thing. What are you seeing? As the virus continues to progress. I mean, is one treatment essentially going to be able to match what the virus seems to continue to evolve, and so that it's just a curiosity of mine, not being I mean, so just to kind of preface this a little bit, right? So I'm not a drug developer and I'm not a virologist at all. And so just a curiosity from somebody, I would say, on the fringe of, the science here.
No, I think that's a great question, and one that I know keeps people awake at night, particularly early in the pandemic where there wasn't a whole lot known about it. But I would say that SARS-CoV-2 is really becoming very much like most viral infectious diseases. Drugs that directly target the virus are going to eventually not work. There
Mm.
will be resistance development that comes up. That is just that comes with the territory. Just as the sun comes up in the east and sets in the west, if a drug directly targets the virus, you're going to see resistance development. So eventually, the drugs that we have now will have less activity as the virus continues to mutate, as we've seen, over time. And this is, the viral evolution is the same thing that happens with HIV. Um, you know, there were drugs that worked very, very well in the 90s that now the the virus can bathe in, and it doesn't do anything. So viral resistance does come up. I think what SARS-CoV-2 has also showed us is that the approaches, particularly when you're looking at anything on the outside of the virus, targeting the spike protein, for example, with these broadly neutralizing antibodies, they'll work for a while, but the virus will eventually develop resistance even to
Mm-hmm.
those. There are several of those antibodies that worked in the very beginning, but now they're off the market because they're no longer active. So, to me, what this means is you need to have a continually evolving drug development paradigm where you're working on new and novel classes of drugs that will help overcome some of these resistance issues. But one of the other advantages to a drug like probenecid, like we're currently working on, is it's directed at the host rather than the target. And so by affecting how the virus interacts with the host and uses the host proteins in its viral replication process, you have a significantly reduced chance of viral resistance. For the virus to evolve resistance, it needs to know what it's evolving against. And so there's no direct viral pressure when you have a drug that doesn't work on the virus. So the virus itself can't figure out how to make, what does it need to change in its genome to get around this particular pressure. And so, and this has been seen with other drugs. Again, I go back to the HIV space. There's a drug called Maraviroc, which is made by Pfizer. It's a CCR5 receptor antagonist. So it targets a human host factor, the CCR5 receptor. It's almost impossible for the HIV virus to develop resistance to that particular compound because there's no direct drug pressure on that virus. And so
Mm,
Maraviroc has been around for 15, 20 years now almost, and there's almost no resistance to it. Now, the problem with that drug is it doesn't work as well as some of the other drugs for a variety of different reasons. But that really, I think, confirms and shows the benefits of a host factor drug that targets that rather than the virus because of resistance development. So I think the virus is going to continue to evolve, and it will eventually evolve into something that the current drugs are not active against. And so you have to continually be working on new drugs, novel targets, because I don't think anybody wants to be in the same place we were 4 years ago, where you've got a virus that all of a sudden nothing is active against.
right. Yeah, I remember every similar to what you're talking about, sort of looking at, what's available on the shelf and, does this
Right.
have any impact, overall and I was active with several of the large pharma companies at that point in time and I just remember them sort of going, okay, so what do we have for, other spike proteins that we can, sort of, use potentially against this thing to try and offer at least some form of solution. And every scientist was combing through data and trying to figure out exactly, what do we do at
Right.
this point in time. So yeah,
Yeah, and that's part of the challenge that a company like us are having, because the drug that we work on targets the host, and so it's a very atypical um, mechanism of action. There's no single target that you can say, okay, this is, you inhibit this particular aspect and the drug's going to work and you'll show activity. And that's a challenge for a large, for any company that wants to do things in a high throughput manner. Because you want to be able to say, okay, well, let's screen all of our drugs against the protease. And we'll find the one that works best against that particular target. We'll do medicinal chemistry if we have to optimize the activity and all the ADME properties, and then again retest it against that particular target. If you get good activity, then you can move forward. That's the traditional approach. I don't want to say it's an easy approach, but it is an approach that there's a recipe for it. You find the enzyme or the target. Maybe you look for an attachment inhibitor, so you block it attaching, to the ACE2, or you block the protease. Those are relatively straightforward approaches that you can screen hundreds of thousands of compounds relatively quickly. The problem with a host factor is you can't do that. You actually have to kind of go back to the basics and do an old fashioned viral replication assay where you actually look at, holistically, does, you incubate this virus with this drug, and then you look at the endpoint of viral replication, the plaques or the colony counts at the end of that, and see if it reduces it. It's very labor intensive. It's time consuming, and that's an approach that just can't be done in a high throughput manner. And so, a lot of the virologists that look at our data are like, well, but it doesn't target these things, and you can only do this particular assay. So, there's questions around, okay, well, does it really work? Um, so, that's part of the challenges when you're working in a novel area, in an area that a lot of people, they understand it because most virologists have done, plaque reduction assays, but they've moved quickly into the higher throughput work. And nobody wants to go back to the old days of,
yeah,
fax machines or modems. And that's kind of an analogy to what we're doing. What we're having to do to test activity is we have to use the modem to dial up and the fax machine to transfer data.
Yeah, I'm very familiar with that aspect because there's a company that I'm currently supporting that's essentially done the same thing. Or like, look, the base sort of science, everybody moved past this and is only looking, now at things that they can use high throughput screening
Right.
for, and we're going to take a different approach because we actually think that we'll find success here. Um, and so, I see that mission, and so I wanted to ask you next, in terms of the overall mission for TrippBio, what do you see that as, and what do you see the future, of the company really being? Do you envision, kind of a broader menu of things and broader focus as well?
Yeah, I think we are. We're very science driven. One of one of the challenges that we've had is because we are doing repurposing. We're working with an older drug. Um, one, in the COVID space, um, that has sort of some negative connotations, as it relates to ivermectin and those types of drugs. And we've avoided a lot of the publicity. We could have gone out very early with some of our data and touted it and really tried to push it, but we didn't want to be painted with that same brush. And so we've taken a very science driven approach. The data that we put out is in peer reviewed publications. It's all, good, solid science. And right now we've got good, solid science around three important respiratory viruses: SARS-CoV-2, influenza, and RSV. So that's our initial focus. We are now, like I mentioned earlier, trying to expand into other negative strand RNA viruses that we're looking at to really try and grasp the full breadth of activity. We know what the mechanism is. We know that there are a number of viruses that use this same mechanism in their replication process. We just don't know yet if they're active or they're susceptible to our compound in that. So we do anticipate potentially moving into those other viruses at funding permitting, but eventually or initially really focused in on these respiratory viruses, because if you look at the ones that we're showing activity against, they're the major respiratory viruses that are circulating in the world right now. SARS-CoV-2, influenza, RSV account for probably 80 to 90% of viral respiratory infections. And so our goal, our vision would be one drug that could treat all three of those viruses. And obviously there's rapid testing that's available, but if we've got good solid clinical activity against those three viruses, you could easily see a scenario where somebody has a respiratory infection, they get a course of treatment with our compound, and they don't even need to be tested because it's been shown 90% of the time it's going to be susceptible to this particular compound. So that's eventually where we would like to be. There's a lot of work between now and then, but we think we've got the solid science to support that vision of the future.
Yeah, and such. I mean, it would be so impactful as well, right? I mean, just in terms of the population to sort of cut across influenza, RSV and COVID. To me, the just looking from the outside, just the impact that one company could have, to deliver some
Right.
some form of treatment for this as well. What are some of the biggest challenges that you've faced in terms of managing the discovery, and then also any, in any clinical programs that you've had, and then how did you overcome them?
Well, I think the biggest challenge is probably the same challenge that every small biotech in the world has, and that's fundraising. raising money. That is the number one challenge. I think a biotech CEO faces, particularly a pre-revenue company like we are, where you're essentially getting money, putting it in a savings account, and spending it down, and you hope you reach some magical fundable milestone before that balance reaches zero. So that's really, to me, the biggest challenge. And it's also one of the biggest creative opportunities because you have a limited amount of money, you have a whole lot of science to do. How do you prioritize? What studies do you do in what order so that you can get the biggest bang for your buck? I know there are companies, Pfizer, GSK, they have billion-dollar research budgets, and a lot of the heads of research there will still say, well, we just don't have enough money to do everything. But the difference is, they do have money, they can do a lot of things. If they have a high priority project, they can shift around things and get whatever done needs to be done. Um, in a small company where you've got limited money, a limited budget, and no capacity to reorganize your priorities and bring more money in to support a program, you've got to be very targeted and very strategic in what questions you ask and how you ask them and who you ask them with.
Mm.
And for us, that's been a real challenge is figuring out, okay, what are the key questions that we need to get answered now? What are questions that can wait 6 months? And then how do we go about financing, those questions and those answers? And that, again, it's the biggest challenge, but it's also the biggest opportunity because sometimes you get some of those questions. We conducted a phase 2 study with our compound, and the goal was to show antiviral potency, um, but we didn't have a lot of money, so we had to figure out how to do that. So we ended up doing the study in India, in the height of the Omicron wave over there, and the study, we weren't able to ask as many questions as I would have liked. I would have liked to have been able to look at cytokine levels and all kind, additional virology, but we had to ask, okay, what's the most important question? Well, what's the viral load reduction? And then an easy question is, what's the reduction in clinical symptoms? So we were able to ask the key questions um, in a capital efficient, time efficient manner, and get the answers. Um, and that we were able to do that for an amount of money that would probably shock anybody that had ever worked in Pfizer or Glaxo, um, because the amount of money we spent on that study was probably the cost of their investigators' meetings. Um, so, and to me, that's part of the fun in working in a small company. Is you have everybody has to do the same thing because we all have the same customer that we're sending our data package to at the end of this, and that's the FDA. So whether you're a big company or a small company, they expect the same kind and quality of data to support the development and approval of these new compounds for these new indications. But how you get there is very different from a company that's got a research budget of $10 billion to a company that has a research budget of $10 million.
Hmm.
Uh, it's there's a lot of creativity that has to go into that $10 million budget that you don't necessarily have to have in the bigger budget area. Now, obviously those companies are creative. They're doing all kinds of things to advance, clinical trial design, wearables, digital, a lot of things are being done to, modernize and really help advance the science along those lines. But in the bottom, the end of the day, it's really about getting drugs into patients, measuring the response, getting the data, packaging it, and sending it to the FDA. There's a lot of different ways to do that that aren't horribly expensive. You just have to be willing to figure it out and do it. And
Yeah.
that's part of what I enjoy about being in a smaller company.
Yeah, I mean I do enjoy the level of creativity that's going on right now. It is, and I've said this a few times in other podcasts as well, that I have to wonder, based on all of the comments that a lot of CEOs have said on this show, will there eventually be a vacuum that all of a sudden we'll just turn around and all these sort of innovations that we're progressing just couldn't anymore, and so there's a lot of companies that stop in the process as well. But I mean, anywhere from some of the more popular things that I can think of would be like Jacob Glanville did pig studies outside of the United States, right? Your work in India certainly is creative as well. And I see a lot of people sort of limiting their questions, sort of saying, hey, look, we think that we could get to an answer here and be able to provide that level of data again, both for investors as well as anybody else that's sort of watching our progress. And it's the only thing that we can do right now because it's the only thing we can afford to do. And you know so it will be interesting to see what gets, I guess, slowed or stopped because of the level of funding that we're seeing
Uh,
as well. But yeah.
yeah.
So the next question I had was, how do you see the future of drug development evolving, especially in the areas of cardiovascular, inflammation, respiratory, and HIV therapies?
Well, I think along the lines of what we were talking about, the number of endpoints, in clinical studies have grown exponentially. The capacity to measure new and novel endpoints has increased. The use of digital tools in clinical trials has really, I think we're just entering that new era of the use of this. Apple Watches can now measure oxygen saturation. You can get an ECG with an Apple Watch. There are lots of things that you can do where you essentially can get continuous data. Um, so I think one of the challenges that we're going to have is harnessing that information in a way that makes it manageable. It's one thing to collect data, um, but you can collect it, but if you don't know how to analyze it and you don't have the capacity to analyze the amount of data that you're getting and understand it, then why collect it? And so I think there'll come a time where we have to make a decision: how much data do we collect? Um, and do we kind of go back and say, okay, well, we just really need to answer the basic questions, because one of the you know I think about I've got some lawyer friends, and one of the things that they've always said, told me is, when you go to a jury trial, you never ask a question of a witness that you don't already know the answer to. Um, so there's a little bit of that, I think, in clinical trials. You have to be careful of the questions that you're asking, because sometimes they can, you can get an answer that it may be meaningless in the overall scheme of things, but it mandates a follow up. You have to, follow that to its logical conclusion, and that can take you down a rabbit hole. And if it's not germane to the primary point, primary objective of the study, it's noise. And I could envision a scenario where, you know, particularly in the cardiovascular field, where these studies are huge. You may need 10, 15, 20,000 patients to show a modest difference in these endpoints. Um, that's a huge amount of data. With that number of individuals, even small differences in some of these endpoints can be statistically significant. And you get a significant response to something, you've got to follow that up, particularly if it's, there's efficacy, um, endpoints that that maybe it doesn't mean anything, or maybe it's in the wrong direction. So you really don't, you really don't know what that means, but you're going to have to follow it up. So I think that's one of the things that we're going to have to grapple with in the future is, how much data do we collect? How do we analyze it? Because if you collect it, you've got to do something with it. You can't just ignore it. And for small companies, you're not going to have that same level of concern because you just can't afford to collect that amount of data. Small companies, can't afford to run a 20,000 patient phase 3 study. So, as the clinical programs balloon, I think that's going to see a real narrowing of, small private biotechs that can do that kind of work. And so you'll see a huge upswing, I think, in partnering, earlier on. And there'll be a lot of investment decisions that get made that shift away from the smaller companies trying to do something novel and unique, to larger investments in the hot spaces.
Yeah.
AI right now is incredibly hot and sexy. So you put AI in your mission statement. It's not a guarantee of funding, but it is a guarantee that you'll at least get a meeting with a VC.
Hmm.
Um, and sometimes that's that's the first step. And that those things are challenging, for CEOs, particularly in the small private space. The larger companies have their own challenges, but in the smaller space, we have some of the same challenges, but ours is one of scale. Again, we're talking, but research budgets that are, maybe tens of millions versus, billions for the larger companies. But you got to raise that money because we're pre-revenue and it's coming out of somebody's pocket.
Right. Yeah. Yeah, and it's I mean it overall does spell a different future, as well for many companies. I do think, on the data side, I mean I just keep running into, more and more brilliant data scientists, and I sort of, see these people, combing through the mountains of data that are getting created, but at the same time, the questions that you're asking, I feel like are valid. Like, how much is enough, and versus, going to that point of having too much and then forcing yourself to do additional things just because of the fact that you've collected additional data. So absolutely critical in your
Right.
journey, from, board member to CEO and throughout your career. I'm sure that you've, received a fair amount of leadership advice, as well. I just would love to know, what's the greatest leadership advice that you've ever received?
Well, I think it's a piece of advice that a lot of folks have gotten. It's the best advice was really around myself, and I should never be the smartest person in the room. If I'm the smartest person in the room of people that I've hired, I've made a mistake.
Mm,
You need to hire people that are much smarter than you are, that are much more capable, um, because if you're if I represent the maximum intelligence in a room, our ability to think outside the box, outside my experiences, is going to be very limited. And so it's a diverse group of individuals that you want with a diversity of opinion, a diversity of experiences. Because what I found is, it could be, someone with, has an offhand remark that somebody else picks up on and says, well, hey, that's an interesting idea. Have you ever thought of this? Well, then somebody else says, yeah, but we did something similar in this past company, and this is how it worked out. But know that if you tweak it a little bit this way, it can change. Well, somebody else will say, well, hey, I've got experience doing that. So it's that diversity of opinion that really, I think, helps move a company forward, particularly when you're working in a cutting edge space, drug development, basic research, where maybe something's never been done before, or something. There are questions about, okay, well, what does this mean? If everybody has the same background and same training, you're just going to get the same responses. You need to bring in those outside folks, and again, people that are much smarter, because the cream rises, and that's really where you want to be. You want to be at the top of the container rather than down at the bottom of the container.
Yeah, for sure. And the way that I've heard that one summarized is, hey, if you're the smartest person in the room, you're in the wrong room. So,
Absolutely,
yeah,
absolutely.
yeah, I love that. I love that as well. So, what inspires you?
Well, I think over the pandemic that has occurred over the last 4 years, there's been a lot of sort of heroes that have come out, a lot of villains as well. But to me, one of the most inspiring stories has been Caitlin Carrico. One of the Nobel Prize winners for her work with mRNA. Her story is just incredibly inspiring because it's she's a scientist, but her story kind of, you can look at it in business terms as well. You know, she did, no one believed her. No one believed in her. But she had an incredibly strong work ethic. She believed in what she was doing, so much so that through most of her scientific career, at least in academia, she never really had lab technicians. She did all of her own pipetting. She did all of her own experiments. You don't get Nobel Prize level scientists that really do that anymore. They run these massive labs with lots of people, churning out brilliant ideas. But here you've got someone that you know rolls up her sleeves, gets into the lab, does her own work. Puts her head down, and just nobody really believes in her, except her colleague, Dr. Wiseman, and they end up with this amazing discovery. Um, and she never started out, I don't believe she ever started out expecting to change the world, or developing a technology that would have, earth shattering effects. But, she developed a technology that saved millions of lives during the pandemic, with, a vaccine for a virus. But that technology also has the capability to do lots of other things. They're looking at, cancer vaccines and all kinds of different approaches with that. And it's just, it's an incredibly inspiring story. And there's a quote from her that I have in a tagline on my signature. And it goes, I'll read it. The history of science, it turns out, is filled with stories of very smart people laughing at good ideas.
Mm.
And, that is particularly resonant for us at TrippBio, where, we've got what we think is a very good idea. Um, and, we're struggling to find that one, smart person that believes us, that believes in the story, and that can help us advance to the next level. Um, but that's I think that goes for a lot of different things in different aspects of someone's life as well.
Yeah, absolutely. What concerns you?
Well, I mentioned it earlier, fundraising.
Yeah.
Raising money is if you read most surveys about what keeps CEOs up at night, particularly small biotech CEOs, it's raising money. Most of us believe strongly in the science that we're doing, so that doesn't really bother us too much. But are we able to raise the money to do the good science that we want? And it's particularly challenging, I think, in infectious diseases. Um, it's that's been an area that has been taking COVID-19 aside, that there really hasn't been a huge focus on investments in infectious diseases, and it's one of the challenges now with antimicrobial resistance. There's just not enough money, basic science money, going into that space, and so that's a concern. And it's you see these stories. Earlier this month, there was a story about a consortium of A-list investors that put together a $400 million round, for obesity. Um, you know, 5 years ago, no venture group was putting money into obesity research. The only ones doing work in that space were the Novo Nordisk and Eli Lilly. And it shows their ability to take these long-term bets. But they were the only ones doing it. But now that they've shown that it works and that there's a market there, everybody is pouring money into this,
Sure.
and it's an obscene amount of money. Um, they're fully capitalizing these companies with very little hard clinical evidence that it's going to work.
Mhm.
They're taking moonshots, but sometimes they work and sometimes they won't. But for a company like ours, you know, it is challenging, to read those headlines and then realize that our fundraising efforts aren't quite as successful, um, and don't have the spotlight on them that they do.
Well, thank you for sharing that, and I do think that I do think that there's just a different level of perspective whenever it comes to just let's say like high tech sort of investments, things that are more attractive based on the spotlight. You hit on it with AI, and I do feel like the spotlight on AI is sort of dimming a little, as well. That there's just not the same level of focus that there was, even just a couple of years ago, whenever you had, you know, essentially tens of millions of dollars, let's put it that way, being at least, thrown at these companies to invest in them. I have a feeling they're fighting a little harder, right now as well, just given the amount of AI companies that are out there. But yeah, it's for sure a tough market. Last question: what excites you?
No, I mentioned this earlier as well. Scientific curiosity. I just, I wake up every morning, especially when I know there's some data coming. Um, Excited. Sometimes I'll wake up, earlier than normal, and be refreshing my email. Okay, when's that email coming? When's that email coming? And opening it, and there's just there's wonder in that. Sometimes the result is great. The curves are so far apart that you don't need statistics to show that they're different. Sometimes the curves are superimposable and you don't need statistics to tell you that there's no effect. But sometimes you know it's a mixed bag, and you've got to really dive into the data and figure out, okay, what worked, what worked, what didn't work, why did it do that, what's the patterns. And I can lose myself in doing that analysis. And there's a story about Einstein that says he enjoyed research so much that sometimes he would look up and realize that he hadn't eaten lunch.
Mm-hmm.
And I can have a real understanding of that, because there are times that I feel the same way. It's like, wow, it's the afternoon, and it was morning when I started. And that's that to me is what it's all about. Is the excitement of the science. Again, asking the question that no one's ever asked before, and when you get the answer, you want to figure it out.
Yeah, absolutely, and it's exciting times for sure. Overall, well, Dr. David Martin, thank you so much for coming on the Life Science Success podcast with me. I greatly appreciate you being here for, explaining to us, all of the great work that's going on at TrippBio, and yeah, I wish you all the greatest success in the future.
Thanks, Don. I appreciate it. I
Yeah,
really enjoyed it.
thanks a lot.